Okay, our next speaker is Dr. Carol Cramer, and she and Dr. Norm Radin are founders of a small company, BioStrategies, and they're using interesting things like plants to try and make large amounts of enzyme for substrate, for enzyme replacement therapy.
So I apologize, or maybe I don't, that many of you may have heard this talk yesterday. But what we would like to do is to talk about approaches of enzyme replacement therapy. If I could have the next slide, please. I'd just like to introduce and say who we are. So BioStrategies is a small company, and we thought we were quite small. This is our entire crew, but we're more than two, so I feel very big all of a sudden. And I would also like to highlight that on our GM1 work, we're working with Sandra Diazzo and Ida Naziata at St. Jude, which many of you know. And at the end, I'm also going to talk about a collaboration that I have with Dr. Chet Whitley that is looking at using this same technology in order to try to target one of the mucopolysaccharidosis diseases. If I could have the next slide. So Dr. Tift gave a great introduction, and after I talked yesterday, I said maybe I should back up a little bit. But I think of a lysosome as the recycling bin. Or you could think of it as the commode. And if you don't have a way to clear this out, your body builds up these compounds. And if I could have the next two little parts. Essentially what happens is that if you can't flush the toilet, if you can't get that stuff out of your cells, it's going to accumulate to really pathological levels, which ends up having significant pathologies of the body. So next slide, please. So what is enzyme replacement therapy? Enzyme replacement therapy is a mechanism whereby you make the protein you need, in this case beta-galactosidase. You make it in another cell system, you purify it, and then you deliver it to the patient, typically by an IV. And often these are given once a week or once every 2 weeks, and for maybe about 7 or 8 diseases now in the lysosomal family, it's very effective. This is a child who's been treated for less than a year, and it really does a lot to correct their visceral symptoms. Next, please. So why is there no ERT right now for GM1 if this is a technology that now is being used to treat many diseases? And if you can hit the bottom button, the clear problem is that all of the Tay-Sachs-related diseases have tremendous central nervous system involvement. And this whole group of enzymes that they use in enzyme replacement therapy have been incapable of getting into the central nervous system, into the brain. Next slide. So our technology involves something we call the RTB lectin. So an enzyme is a protein that does a job. It chops something up. So beta-gal is an enzyme. Okay, a lectin is a protein that binds sugar. And what we have done is made a fusion that puts the human beta-galactosidase protein literally attached as one molecule to our RTB lectin. And why do we do that? Well, it turns out that human cells are completely covered with sugar. They have glycolipids, they have glycoproteins, and so they're like little M&Ms with a sugar coating. And our protein, or our lectin, binds onto that, and they use those sugars to direct it into the cells. So RTB is very good at taking passenger proteins and moving them into cells. And in fact, when it goes into cells, its route is typically going to the lysosome. So it'll carry a lysosomal enzyme and it'll put it in. So next one. Of course, the real question is, can it cross the blood-brain barrier, which current strategies don't. So what do first of all, let me remind you what the blood-brain barrier is. So the blood-brain barrier isn't like a barrier. It turns out it's modified cell walls, the cells that make up your capillaries in the brain have very tight junctions and nothing gets in and out. So they're very selective. And that's to protect your brain. And in fact, what happens is the blood moves through those capillaries, and sitting right outside is something called an astrocyte. For obvious reasons, looks like a star, but they have feet, and basically things that are supposed to go across get passed from the blood vessel into those astrocytes, and they carry them and deliver them to the neurons. So you need to get from inside the blood into the brain parts, and there's a few ways to do it. So small molecules like chaperones actually can sneak between the cells, but that's pretty hard to do in a brain cell because they're very, very tight junctions. What typically people use in all of the current ERTs is something called receptor-mediated transcytosis. So you have protein receptors on the cell surface, and they bind to your protein, and that triggers them to be snorfed in and dragged across the cell. Okay? The problem is those receptor numbers are very low or non-existent on the blood-brain barrier cells. So it might go into your liver, it doesn't cross into the brain. So there's a second mechanism that instead of using a receptor, it just binds onto the surface, and it's, hey, here, I'm here, take me in, and it'll carry across. And that's what RTB functions, is it actually binds and uses this mechanism. The advantage being you're not dependent upon very rare receptors. You're just looking at the sugar coating. So we believe that it can take it in. The next slide. So just to remind you and confirm what Dr. Tiff said, we actually make our proteins in plants, and we do that for reasons of added safety, of productivity, and because our RTB is originally a plant lectin, so it's very good at making that protein. But we've got a cheater way to do it, which is that we put our little gene construct into a bacterial vector, and we basically turn it upside down, suck a vacuum, and we suck the gene right into the leaf. And the leaf takes it into every cell. It says, oh, I'm going to make this, and it actually makes quite a lot of it, and we harvest it 4 days later. So it's pretty quick. We grow greenhouse, suck it in. 4 days later, we can start purifying that protein. Next slide. And that is a very scalable system. So it works very well, and we get nice active protein. Next slide. So very much like the previous speaker just described, we had to show that our RTB fusions worked. So what does work mean? It means it can do a galactosidase. It can chop up galactose off of GM1, and it also means that it binds onto the cell. And so we have a pretty nice assay, and this is a normal set of a normal person's fibroblasts. So those are skin cells, and they're stained so that you can see the nucleus where that DNA is of each cell. So the little blue dots are nuclei. If you do the same study and you do a detection system to look at GM1, which is the substrate you can't degrade, you can see all this white garbage. That's what GM1 patient cells look like, is that they've built all of that up. So we can measure that. We can measure this. So that's the equivalent of that and that. And next slide, please. Next button. So all you need to know really is that here is a patient fibroblast, and if we treat them with beta-gal RTB, our enzyme, you actually can clear all of that at the cell level. So next slide, please. So we've now done this in collaboration with Diazza's group, where we've actually done a short-term administration into the mouse. So we use a tail vein, but that would be the equivalent of a patient getting an IV. And just 24 hours later, when we look at what it looks like in the liver, we can actually see that this is the knockout mouse, which has no activity, no enzyme activity versus wild type. And if we take knockout mice and we give them our enzyme, we can see enzyme activity in there. But more importantly, if we measure the disease substrate, the beta-gal, you can actually show that in wild type, it's very low. Here is a GM mouse looking at their liver, and 24 hours after treatment with our enzyme, you can see that we've really reduced the level of GM1. And the same happens in kidney and spleen. So that suggests that our enzyme gets into the cells, it delivers active beta-galactosidase. It takes the beta-galactosidase into the lysosome where it needs to do, and it actually cleans it up. So that's a very promising indication. But of course, that's liver and spleen, and all of the other ERTs get there easy as pie. So next slide, please. So the question is, does it get to the central nervous system? And so this is a little bit complicated, but we've looked at two parts: the cerebellum, which sits right here in the back of the brain, and the spinal cord. And one of the reasons that we look at the spinal cord is that one of the issues with the disease is that sometimes they can correct parts of the brain, but because the nerves going down the spinal cord have been destroyed, you don't necessarily correct the physical parts of being able to move and ambulate. So this is looking in this case at the presence of beta-galactosidase by using an antibody and looking at total proteins. So even though the wild-type mouse has a lot, we can, after a period of time, still see that beta-gal has gone in. This was now given to the mouse 4 times, so twice a week for 2 weeks. So again, this is not a long-term study. But you can see that the disease mice, the GM1 gangliosidosis mice, have tons of this stuff in there. And after just 4 administrations, in one mouse, we've got it almost down to wild type. So you can see that there's a variation, but clearly it's getting into the brain, which means it's crossing the blood-brain barrier. We also could see that same sort of some correction in the spinal cord as well. And remember, this is still an ERT you typically treat over a long period of time. So this is short-term correction. But again, that looks very, very promising. Next one, please. So just to summarize, based on these short-term studies, a little bit long, but 2 and 4 weeks, we know that our beta-gal RTB fusion crosses the blood-brain barrier and can actually get into the brain and reduce the GM1 substrate. So we think that that's very promising. Next slide. So I also wanted to report that in collaboration with Chet Whitley's lab, we've been looking at another mouse disease model, the mucopolysaccharidosis 1, or Hurler mouse. And I just bring that up because they have tested our molecule over a longer period of time. So they've given it weekly for 8 weeks, and which in that mouse model takes it long enough to see whether or not you can get changes and impacts on behavior. So there is something called a Barnes maze, where you drop the mouse in the middle, they're under a bright light, and their instinct is to run and hide. And you do this over multiple days, and you see how long it takes them to find the one hole that is open. So the mouse races across, it tries to find it, and it goes in. And you do this repeated times over 6 days. And so you see whether or not it learns, and the idea is that it actually goes faster. So in this case, you can see that the Hurler mice, so these are the sick mice. Did I lose this? Oh. The sick mice actually become less capable of quickly learning. So this axis is how much time it takes you to get to the hole. And you can see that over multiple days, they don't get much better. They learn some and then they plateau. The gray line in here, I think, yes, the gray line, which is right here, is actually the normal mice. And we were very excited when Chet sent us the data that basically showed that both of our treatment groups actually corrected that behavior. So for us, so this isn't with GM1, we're in the process of developing those long-term studies with GM1, but this suggests that our carrier clearly gets to the parts of the brain and to places that will help in correction. So overall, we're very encouraged about this, and we basically feel that we now have, from a couple of models, a real incentive that this lectin will carry into the brain. It seems to deliver in all of the tissues that we have looked at so far, and it clearly corrects protein across the blood-brain barrier in a way that makes it do its job. So we're very enthusiastic about that. So we're encouraged, and so the question is, what are our next steps? And they're not a lot different from everybody else's next steps. Some of us are further ahead and further behind, but essentially one of our goals is fairly quickly to meet with the FDA to make sure that the studies we do are the studies they want us to do. And that's called a pre-IND, which is before the pre, and then the IND, which is actually the ability to go into clinical trials. So for clinical, we're now going to expand the GM1 studies to do very much like the Hurler mouse, of going on, making sure that when we look in the brain, we've cleared the pathology within the neurons, and also that we correct the behavior. We'll need to do dose-finding studies. Again, in our experiments with Dr. Whitley on the Hurler mouse, one of the things that was very encouraging as well is the same dose that they currently use in Hurler patients that they found corrective, and that's based on the amount of protein per kilogram of body weight. That same dose actually worked in the mice with our RTB carrier. So that's actually encouraging as well. But we'll need to do dose-finding studies. We'll need to do toxicity studies to make sure that this molecule is safe. And again, one of the things that is encouraging is that the enzyme replacement therapy is widely used in these other diseases. So it's not like we're breaking huge new ground from the point of view of the FDA. And then on our side, we have to scale up this production. So right now we're a little company, and we literally grow our plants in, 2 rooms that are the size of 3 of those tables. So we have to get this up to really being able to grow a lot of this stuff and do some formulation, and then we need to apply for clinical trials. So next slide. So in summary, we really want to thank Tay Sachs that really hit us with that first study and accelerated all of these studies in looking at the GM1. And just to say that our group is very committed to trying to bring this ERT for GM1 forward. So I'll answer any questions if you have them.
Was wondering about maybe a timeline for the things you just mentioned on the last slide.
Could you go back to the last slide? So our expectation is, with appropriate funding, that we could basically mobilize this in the next 2 to 3 years. So if we started right now, potentially 2 years, we have some grants funding that wouldn't kick in, but we're thinking that we could do this in 2 to at most 3 years. And you can't ever figure out, as we've seen repeatedly at this meeting, the pace at which the FDA will move things forward. But I think there's a couple things going for this. One is that ERTs aren't new. 2 is that there's nothing for these patients at this point. And I think FDA is becoming more and more cognizant of their obligation to try to move things more quickly.
Other questions?
As far as what you're saying, as far as the mouse trials and things, you were saying 1 to 2 week testing that you did with them, correct? And this is not a question just for you. This is more for, I guess, everyone else's knowledge with all of you guys. 2 to 3 years. Why can't I just stand on the doorstep of the FDA and like start knocking people out? I'm seriously not kidding. Why couldn't this have been available in February before I lost my daughter, or anybody else in this room? And it's not just yours or yours.
Yeah.
I love the work that you guys are doing, but can someone please tell us how to make these things happen?
How come I was the lucky one to get that question? No.
Actually, I was going to go to him first, but
actually, shall we say we feel your pain. And among the things that, I mean, we have many steps to go through. We don't have the capability at this moment to make enough making enough for a mouse is quite different. So we do have an obligation. What would be the minimum that I would ethically feel comfortable with? We need to be able to make this compound at scale. We need to do toxicity so that we don't end up, making a big mistake. Sort of that. And once those things happen, when I said the FDA is really pushing, they have a number of new options. They have something called an individual IND, an IIND, where for compassionate use, you have the ability to accelerate and test things. They have fast track and a variety of mechanisms where they recognize the challenge for rare diseases, and they're trying to accelerate. So we can't necessarily control that. On the other hand, do we think it makes a difference for patient families to approach FDA and the government and NIH and sort of continuously let them know what a difference it means? That does make a difference. And I think that the patient advocacy component, and there's actually groups that will walk you into your senators, will coordinate for the families to go visit their senators, and that has actually made a difference. So the way, we're a small company, the way we feel it's important for us to go forward is in partnership with the foundations and the patient groups, with NIH, with our company, and to mobilize as quickly as possible. And even though I don't have a child with one of these diseases, I recognize when we first started doing this, we went to our first mucopolysaccharidosis meeting probably in 1994, and they were putting the Hurler enzyme into beagles. And a parent got up with tears literally running down her face, and she said, do you know how, thank you so much for your work on our behalf. Do you know how it makes us feel as parents that you're taking the human enzyme and you cured that dog, and my son is sitting here and will not survive until you have ability to put it into humans. So I think about that every day, and we understand how hard that has to be.
Trust me, I did not mean that in a degrading way at all to anybody that's working on this.
Yeah. No. I recognize that. We do feel your pain,
And I think
we're frustrated as well.
I think part of the issue is it has to be done carefully enough that we choose the right measures to show success. Because if you don't choose the right population and the right measure, and you don't achieve your outcome measures, you go down in flames, and then nobody gets it approved. So there's, I get it. Believe me, I get it. But you have to pick the right things. Otherwise, you're done before you start, because they'll never approve it.
Once you start the clinical trials, do you anticipate the patients regaining skills?
Do you want to answer that?
We don't know. And you knew I was going to say that. Generally speaking, one would think, and I had a conversation with a parent at lunch, one would think that the children who have progressed least far might have the greatest benefit. But we don't know, because we look at kids who have lost a lot, for example, a lot of brain volume, who can still do a ton of things that I would never expected them to be able to do. So I haven't got a clue, to be honest with you.
Yeah, and I guess I might answer that just from the point of view of some of the mouse trials. Again, depending upon how early you start, some of these therapies that you start already in adult, and you can actually see significant improvement, even in learning behaviors. So again, early is always better, and Dr. Tiff's sort of plea to figure out ways to diagnose these children more quickly is really going to be important. But there is evidence that you can get significant correction even with light stuff. These ERTs or chaperones will not restore dead neurons or even stimulate them to grow.
And we
So,
learn how to make more.
yeah.
Okay. Thank you very much, Carol. That was great.